Welcome to the 81st Annual Cold Spring Harbor Laboratory Symposium on Quantitative Biology. This year's topic is Targeting Cancer. I'm Elizabeth McKenna, an Associate Editor from Cancer Discovery, and I'm sitting here today with Dr. Daniel Haber, the Director of the Massachusetts General Hospital Cancer Center. Welcome, Daniel.
Thank you.
So this morning you spoke at the symposium on your work on circulating tumor cells. Can you maybe briefly introduce the topic of circulating tumor cells for the audience?
Sure. Circulating tumor cells are cancer cells that are found in the blood. And this is how cancer spreads from one organ or from a primary organ to give rise to metastases.
Mm-hmm.
The challenge has been that these cells are really rare and they're surrounded by lots and lots of blood cells. But if you can find ways of isolating them, then the biology is really fabulous and really fascinating. So we've spent a lot of time developing a number of technologies that are better and better, and ultimately now we have very good technologies for pulling out these cells out of the blood, and then you can study both metastasis as well as cancer progression.
Can you discuss some of the techniques? I mean, you've been a pioneer in various methods, and maybe discuss the different ways you can isolate circulating tumor cells, and perhaps which method you think is the best out of the different ways.
Sure. There are a number of different techniques. Some have been around for a while, some are more new. Some techniques use the fact that cancer cells are somewhat larger than blood cells, so they try to filter them. But some cancer cells are not that large, so you miss a lot that way. Other technologies involve trying to use antibodies against epitopes on tumor cells or on epithelial cells and pulling them out. And those have been kind of standard in the field. The challenge is that some epithelial cells become more mesenchymal, some cancer cells have different epitopes, so it's hard to get a reliable antibody to always pull out tumor cells.
Mm-hmm.
So the technologies that we've used, and this is in collaboration with Mehmet Toner, a bioengineer at Mass General, has been to try to purify out the normal cells. So if you could only have an antibody against white blood cells, and you could get rid of red blood cells, and you get rid of everything that's normal, you're left with the cells that don't belong in the blood, and these are the tumor cells. But to do that is hard, and you need a technology that is really, really good and efficient at purifying out cells. And that's this whole field of microfluidics, where you can line up the cells in such a way that they pass single file in a channel. And if they do that, then they have a little magnetic particle attached to an antibody. You pass them through a magnet, and then you can separate them. So it's really a place where the technology has gotten more and more complex, but more and more powerful. And finally, we're now at the time when the technology is good enough that you can reliably pull out these cells and ask the questions that we've always been wanting to ask.
So what are some of the characteristics of circulating tumor cells? Are they very similar to the parental tumor, or have they diverged in any way? Like, how are they generated, and which ones are selected for metastasizing?
It's an interesting question. Clearly there's a lot of heterogeneity. The vast number of circulating cancer cells in the blood die. Mm-hmm. And in fact, one of the interesting things is we find that early cancers can send out cells into the blood way before any metastases occur, and presumably most of these tumor cells die. So when you capture cells from a blood sample, maybe 90% or so may be cells that are destined to just die, but some of them may be alive, and some of them may be endowed with the potential to give rise to metastasis. Mm-hmm. So you're looking at a heterogeneous population. Mm-hmm. Recently we've been able to culture some of them, and the ones that grow out in the lab will look different. They grow in clusters, they grow under hypoxic conditions, they're very, very tumorigenic when you inject them back into mice. So there's clearly a subpopulation in the blood that are bad guys and bad actors, and those are the ones that are in some ways the most interesting to study.
Mm-hmm. So what are some of the things that you've been able to learn about the original tumor from the circulating tumor cells, from either collecting them or culturing them? Can you sequence that, do DNA sequencing, learn anything about the development of the tumor from them?
Sure, pretty much anything that you can apply to a tumor, you can start to be able to do with circulating tumor cells. So you can certainly stain them for all kind of markers at the protein level or at the RNA in situ level. Mm-hmm. You can pick individual cells and do RNA sequencing or do whole genome amplification and look for mutations. The technologies have to be adapted for minute amounts of material, but in a way those technologies are available now, and you can access heterogeneity of circulating tumor cells. In a recent study, we're looking at advanced prostate cancer, and we could see how even within an individual, different cells had different markers and different genetic abnormalities. So it's a helpful way of looking at the complexity of cancer. But the most important thing is that you can do this non-invasively in a blood test, and you can do this over and over again. And increasingly now we have these effective treatments for cancer, and the more effective your treatment is, the more the cancer shrinks, and when it grows back, it has different properties. And you're not going to be successful at treating it a second time with the drug that you used the first time. So cancers evolve, and you can rebiopsy patients and rebiopsy their tumors, but you can't do that over and over again. So that's the great potential of these liquid biopsies, is that you can do this every week if you need to, and try to see how the cancers evolve over time.
Right. So that leads me to my next question. Can you talk a little bit about how analysis of circulating tumor cells is moving towards being used in the clinic, and what are the important questions that need to be addressed to demonstrate that this is going to have clinical utility in the liquid biopsying, as it's called?
Sure. I mean, there are three different technologies, all at different stages. We have circulating tumor cells, where you look at whole cells, where you can do anything from culture them to look at protein, RNA, or mutational changes. There's plasma DNA, free plasma DNA, that you can analyze, which is primarily looking at mutations and using that as a surrogate for what the cells do. And then there are exosomes, which there's still technologies to try to isolate those, but they have little collections of mostly RNA, but other things as well. So ultimately, I think in the clinic there will be different applications for each one. There's some things that you'll do with one, some with the others, depending on whether a particular cancer has recurrent mutations or has RNA changes, or what you want to ask. I think in terms of applications, the most obvious one is when you give a treatment and the tumor shrinks but then comes back, trying to figure out what's the mechanism of resistance. And we're now at a place where there are second, third, fourth generation treatments, and you want to guide those based on the mechanism of resistance. So if blood-based biopsies will give you a sense of the mechanism of resistance, you'll know how to treat the patient. But as that becomes more and more applied, I think we'll be applying this earlier and earlier. There are many times when the primary biopsy isn't so good, or when it takes time to get a primary biopsy. So you could see that moving earlier in the course of treatment. I think ultimately what we're also discovering is that some of these cells in the blood have different properties, and that raises the question of different targets. In fact, there's a lot of interest in why tumor cells in the blood may be more sensitive to different stresses, different oxygen stressors, or loss of anchorage dependence. And all of a sudden we may have new treatments for cancer based on suppressing their survival in the blood. That would be really exciting.
So are tests for circulating tumor cells or cell-free DNA, are they being incorporated into clinical trial design? Like what bar needs to be met for this to be really introduced into clinical practice?
I think both ctDNA, plasma DNA, as well as CTCs are involved in a large number of clinical trials. I think that to really gain acceptance and to really be part of clinical care, you have to have a demonstrated benefit, and then you need some kind of reimbursement structure that will make those viable. I think that's another challenge of the field of cancer treatment, is that as our treatments have gotten better and better, the diagnostics becomes more and more important to guide those. But the way that we support both research and application of diagnostics hasn't kept up. So in some way we need to put some more effort in supporting diagnostics so that we can direct those therapeutics more effectively.
Is there a particular disease that you see these liquid biopsies changing practice in the near-term future? I'd
say the two. One is definitely where you have targeted therapies, and they give rise to a couple defined mechanisms of resistance. So if you have mutation A, you need this treatment. If you have amplification B, you need that treatment, and it really makes a difference. So I could see that happening relatively quickly, and that's lung cancer, potentially melanoma, breast, and so on. There are other cancers where when it spreads, it goes to bone. So that's prostate and often breast cancer. And when metastatic cancer goes to bone, it's very hard to biopsy the bone. So you're treating patients with a fairly indolent disease where there are lots of medical choices and you don't know which one's the right one. And that's where blood sampling would be really, really helpful.
So moving back to the biology of circulating tumor cells, what do you see the next big unanswered questions in the field being?
I think we're going to definitely see monitoring of cancer. We're starting to see new pathways that are relevant to circulating tumor cells that we hadn't appreciated before based on just tumor analyses. So the idea of whether there could be some therapeutics that would target CTCs is really intriguing. My little obsession, if you will, is to move the way that we diagnose cancer at an earlier stage. If you can diagnose cancer when instead of 1 trillion cells, there's only 1 million cells, any kind of treatment option that you have would be so much more effective. You'd have less resistance, and any kind of cancer would be more sensitive to therapy. The challenge is that most early diagnosis of cancer is fraught with risk.
Right.
You know, you pick up non-invasive cancers, you have false positives. I think that blood-based diagnostics is going to have a major effort to contribute here. Because if a cancer cell invades into the blood, it's unlikely to be an indolent cell.
Right.
So if you can combine blood-based diagnostics with what we currently have for early detection of cancer, I think we could have a really big impact.
So you think that the presence of circulating tumor cells will occur before any change that would be detectable by imaging per se? So you could use it to track the disease before it might be detectable otherwise, then, right?
I think there'll be a combination of assays like that, but for example, in 20 patients with localized prostate cancer, we could find circulating tumor cells in about 1/4 to 1/5 of them before surgery. And after surgery, they were gone. And none of these patients have relapsed with 4 years follow-up. So it really tells us that a subset of patients will have tumors that send cells into the blood.
Right.
And that offers a diagnostic opportunity. And it may be the same in lung, in breast, in other types of cancers as well. So it won't be every patient, and you might have to combine it with whether it's a mammogram or a PSA or a lung screening, but it's a whole new dimension of diagnostics.
So are there ongoing clinical trials to see if the number of circulating tumor cells correlates with response? I mean, do you need large number of patients to make it that it's a clinical benchmark?
I think that's the challenge.
Yeah.
When you have a patient with cancer who's being treated, that patient is their own control in a way, and you don't need that many patients to see if there's a change.
Right.
If you're looking at early detection, even in high-risk individuals, the number of patients you need to study or the number of healthy individuals at risk you need to study is considerably higher. So you have to design those studies very carefully. But you're absolutely right. It's a larger number of patients, and the assays have to be much more sensitive and much more reliable and robust. And again, you're entering the stage where you're entering a field where you don't want to make mistakes.
Right.
You don't want to be testing people and coming up with false positives. So it's a whole new dimension. But ultimately, it could have a tremendous impact on the entire field.
That's great. Really interesting work, and thank you for joining me, and thank you for coming to the symposium. And I think that's all we have time for. So thanks very much.
Thank you very much.